So my name is Victoria Laye, and I'm a graduate student at IMET. I work in the DasSarma lab, specifically with psychrophilic archaea. Psychrophilic means that they're able to grow at low temperatures. So specifically, my organisms are able to grow as low as 0 degrees Celsius. So by understanding how psychrophilic organisms and their proteins function, we're able to get a better picture of how proteins are able to function in the cold and can then use that information to make other proteins that we use in industrial practices, like for pre-digesting baby food or taking the lactose out of milk for lactose-free milk. We can make those processes more green because if it's able to function at a lower temperature, you don't require as much energy to be put into the system. No one else is doing cold active protein work in the way that I'm doing it. So I was sort of on my own. I mean, obviously I had a lot of guidance from Dr. DasSarma. Once you work through all the kinks, it starts to go smoothly and it's just so rewarding. We specifically use Halobacterium species NRC1, which is very easy to break open so that we can get the protein or the plasmid or whatever it is we're trying to get out of the organism, because it requires such high salt. So you can just add some water and it breaks right open. Then once we have the plasmid in the organism, I actually have to get the protein from there. So the protein that I specifically work with is beta-galactosidase. And because of that, it has a very simple assay to figure out whether it's working or not. It just cleaves the ONPG into ONP, and ONP is yellow. So essentially, you add protein, you add ONPG, and all of a sudden it turns yellow. So then you know it's working. And I can use a spectrometer to measure how quickly the solution is turning yellow. So that gives me an idea of how well the protein is working under different conditions. It's a very lofty goal to be able to define what it is that makes a cold adapted protein able to function in the cold, whereas other proteins are not able to do that. But I hope to at least get close to that to give some sort of framework for what makes a cold adapted protein cold adapted. Working with cold adapted enzymes and cold adapted organisms in general, we're looking at what are called the building blocks of life. But by doing that, it gives us a better understanding of what we can do next. It gives us a good foundation so that we can look at things like greener technologies, where we can make enzymes work the way that we want them to work. We can essentially bend nature to our will. The only way we can build on what we have is if we have that solid foundation, and that's what my research hopefully will give us, a solid foundation to build off of to create these greener technologies and be able to build a better future.